welcome all the attendees on behalf of the National Academy of Sciences India Delhi chapter and MHRD-Institution Innovation Council, Deen Upadhyaya College, University of Delhi. Under the aegis of DBT Star College program, we are organizing this technical talk. And today we have Professor Brian from University of Illinois, and he'll be talking about finding needles in a haystack, liquid biopsy for nucleic acid and protein molecular biomarkers for disease diagnosis. And as a customary, I'll introduce Ryan, who has been a faculty member in the Department of Electrical and Computer Engineering and the Department Bioengineering at the University of Illinois since 2004, following a 15-year career in industry. His technical focus is the utilization of photonics for biosensing and applications that includes life science research, diagnostics, environmental monitoring, and pharmaceutical screening. He has over 85 issued US patents and over 180 peer-reviewed journal publications. He is a fellow of NAS, IEEE, Optical Society of America, Royal Society of Chemistry, AAAS, and AIMD. I welcome Brian for delivering his talk.
Well, thank you very much, Professor Saxena, for the very kind introduction and the opportunity to share some of the work that my colleagues and I are doing here at the University of Illinois. And also, very kind thanks to National Academy of Sciences of India, again, for organizing this and providing a venue to communicate with scientists in other parts of the world. Very grateful to all of you for coming, spending part of your evening learning about biosensors. Here in the University of Illinois, it's a beautiful day today, kind of hard to tell all the crazy things going on in the world and what an extraordinary time that it's been. But I hope that you're all doing well, staying safe and healthy, and I just wish you all the best. If you do have a chance to come visit Urbana, Illinois, we're in the central part of the United States, a very nice college town of around 150,000 people, surrounded by corn and soybeans for many miles in all directions. Our nearest city is Chicago, for some of you who may have had a chance to visit there. Also, before I start, I'd like to say a special hello to Priyash Barya. Priyash is a student from Birla Institute of Technology and Science, who's joining my research group here at Illinois in the fall semester. So I'm really grateful to have a great new student from India come join my group in the fall. So the topic for my talk is discussing how to detect molecules at low concentrations, among many other things that are in clinical samples, which is very much like looking for a needle in a haystack. And before I give my whole talk, I'd like to acknowledge that the things that I'll tell you about today are highly multidisciplinary and really represent the efforts of many people, not just myself. And so I mentioned the graduate students and postdocs in the nanosensors group pictured here on the left. But also, I've been fortunate to have really great collaborators that I'll mention throughout the talk that bring their own expertise to these projects in bioengineering, nanoparticle engineering, molecular biology and chemistry, and then clinical applications. And we were very fortunate to get funding from the US National Science Foundation and the National Institutes of Health. So I'd like to describe the work that we're doing in this framework that we've created here called the Center for Genomic Diagnostics, which is an effort that's a team science approach to liquid biopsy that I lead here at Illinois at our Institute for Genomic Biology. And the basic idea here is using the liquid biopsy techniques to reveal things about what's going on inside our bodies. And so many of you know that, of course, genome sequencing has opened up a new era in biology, and that genome sequencing tools are very powerful. But nonetheless, they're expensive and difficult to use. They require expensive equipment, very knowledgeable technicians, quite a lot of data analysis. But genome sequencing reveals a lot of information about presence of mutations, the transcriptome. Even now, genome sequencing can be applied to unique tissues and even to single cells. And all that genome sequencing information is revealing a lot about new biomarkers that can become the target for biosensing detection. And so within our center, we're utilizing genome sequencing information, but knowing that genome sequencing is something that can't be done routinely, we want to combine that with liquid biopsy techniques where you can extract a few droplets of test sample from a patient, perhaps a few droplets of blood, and have tests that can be performed frequently. And they may not, within that simple test, might not be able to know everything about the whole genome, but nonetheless, you can target specific biomarkers, and liquid biopsy samples, like a few droplets of blood, can contain a wealth of information through the molecules contained in it. And so these can be nucleic acid molecules like microRNA, messenger RNA, circulating tumor DNA, circulating free DNA. And I'll describe things called extracellular vesicles that we use as a reservoir of molecules. But with our liquid biopsy techniques, we seek to be able to test non-invasively, inexpensively, and frequently so as to have a clinical impact. And so the idea here is that the molecules you can detect from a liquid biopsy can reveal things about health and wellness of a patient. There are now biomarker molecules that are revealed to give information about health and wellness, like nutrition, aging, exposure to the environment. But also, as physiology goes from normal to disease states, of course, liquid biopsy tells us things about health status having to do with diseases like cardiac disease, sepsis, inflammation, infectious disease, and cancer are among those most commonly used as methods biomarkers can be detected for diagnostics. But information from these tests can be used to feed back to our biomarker analysis, tell us which biomarkers are most informative, have something to do and correlate with state of health and wellness or of disease. And then, of course, we want to use these tools for disease diagnostics, ways to inform whether or not drug treatments are having a desired effect, and even as life science tools to understand human biology at a more fundamental level. And so within our university and with collaborators, we're developing this pipeline structure to start with genomic and proteomic analysis, which just generates enormous amounts of information that can be studied using computational bioinformatics tools. Those tools can be used to reveal novel biomarkers that can become the targets for our detection. But myself and many of my colleagues are engineers and chemists. And so we can, once we know the target that we're looking for, we can combine that with ultra-sensitive detection technologies, ultra-selective biochemistry and molecular biology methods that enable us to do different things. So we may be interested in having sensors that can be used at the point of care, or multiplexing to detect lots of targets at the same time in a single sample. As engineers, we're great at developing microfluidic devices that can automate parts of the sample preparation, give us the ability to automate things that typically technicians would do with pipettes in the laboratory. And so with all those engineering things, we're developing intellectual property. We collaborate with clinicians who provide us with a lot of biological knowledge and clinical knowledge about how our biomarker tests can have an impact on healthcare. But also they can give us sample repositories from their patients where we can select people that have different health status, whether aging, cardiac disease, cancer, and so on. And so we can use those to first help us to develop the technology, but then use it in the context of clinical studies and technologies that can be used for helping clinical practice. And our big goal in life is to translate these technologies to actual clinical practice by licensing it to companies, starting companies, getting research grants, things like that. So there's a large cohort of faculty from many different disciplines that are participating in all of this at the University of Illinois. And so I just list them here. You can kind of see all their different disciplines. And then we have several organizations through the campus, such as the Illinois Cancer Center, the Institute for Genomic Biology that I already mentioned, something called the Discovery Partners Institute, which is being founded in Chicago with a biomedical translation facility to help technologies become commercialized. College of Engineering, the Micro and Nanotechnology Laboratory, which is where my lab and office are, and several other organizations, including collaboration with Mayo Clinic, a big clinical research hospital in Minnesota. And then, of course, there's a lot of computation involved in all this, and we're fortunate to have a really strong National Center for Supercomputing Applications that deals with the computational bioinformatics aspects of this. So I know it's a lengthy introduction, but I really wanted to show you the context in which these projects are coming from, that we've been working with many people for several years to build this whole infrastructure for doing biomolecular liquid biopsy. So with that, I can jump into the talk. So I think many of you are already familiar with what a needle biopsy is, and where a needle is inserted into the body to withdraw a plug of tissue that's analyzed by imaging and pathologists, where a needle biopsy is, of course, invasive, painful procedure, can't be performed very often. And so we're interested in less invasive methods that might even be more informative than a needle biopsy. And we call this a liquid biopsy. And as I mentioned, the idea is that a droplet of blood can contain many molecules that can tell you a lot about the person as cells and tissues throughout the body will release material into the circulation. Liquid biopsy has been in the news quite a lot in a negative context in the past couple of years. I don't know how many of you have heard of a failed company called Theranos that gave liquid biopsy a bad name with a failed effort that raised enormous amounts of money in Silicon Valley, just generated a huge amount of interest and selling the promise of liquid biopsy, but not able to deliver. And so if anything can be learned from the lesson of Theranos, you can even watch it on HBO or read an excellent book about it. It's that they tried to use conventional technologies to perform liquid biopsy on very small volume test samples, but failed. And so the lesson that I would draw from this, among many, is that new technology is needed to make liquid biopsy a reality for clinical practice. So I wanted to describe one type of source of molecules in blood that's very useful reservoir of material, and these are called exosomes. So exosomes are lipid, like spherical lipid bubbles that are released from cells that take part of the cell membrane away from the cell with them, along with some of the cell's internal contents. And so cancer cells release exosomes. Healthy cells also release exosomes. But once the exosome is released from the cell, it can travel throughout the body, circulating throughout. You can bet for sure that exosomes will be available at a very low concentration, but the molecules inside them reveal a lot about the genomics and proteomics of the cell of origin. So exosomes contain on their outer surface proteins that can be used to capture them and even identify what type of cell they came from originally. But then, beautiful thing about exosomes is that they protect their internal contents from enzymes and things in the blood that would ordinarily degrade molecules like nucleic acids. And so by capturing exosomes, extracting them from blood, breaking them open, now you can have microRNA, messenger RNA proteins that are representative of the cells they came from that were protected from the other things that can be found in the circulation. Exosomes are so small that they can permeate through tissue, and they can be found in not only blood, but also in urine, saliva, spinal fluid. All these bodily fluids have been found as sources for exosomes. So in my talk today, I wanted to focus on one a specific application for detecting molecules that come from exosomes. And this will be in the context of genomics-guided cancer treatment. So I'll discuss prostate cancer as a specific example, but you could apply the same methodology to many other types of cancers. In this context, we're seeking to detect biomarker molecules that are available at very small concentrations. And molecules themselves are small. And so we developed a methodology for detecting individual molecules and digitally counting them using some tricks of electromagnetics and a technique that we invented called photonic plasmonic resonator hybrids. So I'll tell you what that is. And that's really like the fundamental thing from an electromagnetic standpoint that we're using to make small things like nanoparticles look big. But we've used that electromagnetics concept to develop a new type of microscope that allows us to visualize and count individual molecules. And so what we call this microscope a photonic resonator absorption microscope, or PRAM. So I'll tell you what PRAM is and how that works. And then using this form of microscopy, in collaboration with Yi Lu, a chemistry professor here at Illinois, we developed a new type of assay that we call activate, capture, and digital counting, or ACDC. And we've applied it in a few contexts, but for detecting microRNA biomarkers for prostate cancer. We've also used it for detecting proteins as well in the context of HIV viral load monitoring. So that's really the context of my talk today. If I have a little time left at the end, I have a few new things to also show you, but we'll just kind of see how the time goes as I get to about half hour from now. So the application that we have in mind here, we're focusing on metastatic prostate cancer. And prostate cancer evolves from some cells in the prostate gland that have developed a whole series of genetic mutations, and those genetic mutations are present in the cancer cells and not in healthy cells. The cancerous cells release exosomes into the circulation, and those exosomes contain a nucleic acid molecule called microRNA. Where microRNA is a nucleic acid, has a sequence of nucleic acid bases that have a sequence that's only present in the presence of the mutation. And so there are many microRNA molecules, but only specific ones that carry the mutation. And so we're interested to detect those, since those mutated microRNA sequences tell us something about the presence and the severity of the cancer by measuring their concentration. So we're interested to detect these microRNAs, but also to quantify them. So just to show you clinically how important these microRNA molecules are, this is some data from my collaborator, Manish Kohli, a prostate cancer physician at Huntsman Cancer Institute in Salt Lake City, Utah. What's shown here on this graph is survival as a function of months, where every time this curve goes down by one increment, that represents a person who passed away from their prostate cancer. So greater survival is represented by a chart that goes off into the horizontal direction, and poorer survival represented here by one that drops off rapidly. Well, what's shown here is how survival is linked to the concentration of two specific microRNA sequences. They have very uninteresting names. These are called microRNA 375 and microRNA 1290. But you can see here that when their concentration is low, survival is better. If their concentration is high, survival is more poor. So even just being able to differentiate high from low concentration of these microRNA molecules can tell a physician something about how aggressive someone's prostate cancer is and whether the physician might go for a more aggressive form of chemotherapy or radiation treatment. So we set about doing a study to gather blood samples from prostate cancer patients every day during their chemotherapy. We were interested to see whether these microRNA molecules changed concentrations and how much they changed as a function of their chemotherapy treatment being successful or not successful. And so we gave the patients these kits called hemospot kits, where they put a couple droplets of blood into the center of this spiral paper strip every day. The droplets would go in and follow the spiral path around the perimeter of the cartridge. And then small things like the microRNA in exosomes would be able to travel a greater distance than big things like red and white blood cells. And so the patients would put a few droplets of blood into the HemoSpot kit, seal it, and then mail it to the University of Illinois, where we opened it up and we punched out different places of this paper where the exosomes were found to reside. And then we analyzed them using the conventional laboratory technique called QRT-PCR for polymerase chain reaction. What we found is that these, here just looking at microRNA 375, here's a patient whose microRNA 375 concentration varied by about 4 orders of magnitude over the course of only 8 days of chemotherapy. So you can see how this is something that the clinician would like to not see. That this concentration going way up might represent that the chemotherapy being applied is not very successful. We saw other patients where the microRNA 375 stayed around the same, others where it went down, but we saw huge amounts of variation even over the course of a few days from patient to patient. This type of measurement, the RT-PCR, is very tedious. It's very difficult to quantify the amount, and it took one postdoc, a very talented postdoc, many months to measure, I think it was 15 patients, 8 samples per patient. Just to measure all those samples took many, many months to do. And so it was an informative study, but also not something that we could do on a sustained basis for like therapeutic intervention. So our goals then are to detect these molecules, but we have to do so over several logs dynamic range of concentration, where the low level of concentration extends down into the femtomolar, like 10 to the minus 15 moles per liter level. So we have to detect very low concentrations, but also very high concentrations and quantify the difference. We're interested in an approach that could have an inexpensive detection instrument, even one that could be used in a doctor's clinic, and also an approach that could be very specific to detecting the tumor-related microRNA sequences, but not all the other ones. And then we'd like this type of test to be as simple as possible, even one step if possible. And I'll show you how we've been managing to do that. This low concentration problem is something that is not new, that there's been many people from many disciplines seeking to detect low concentrations of analyte in a complex test sample. The approach that I like to think of as the chemist's approach is is enzymatic amplification. This is exemplified by polymerase chain reaction or enzyme-linked immunosorbent assay, where you start with detecting a single molecule, but you have some type of enzymatic reaction that turns one molecule into many, many millions of molecules. And if you wait long enough, you can generate millions of molecules out of a small initial group. So chemists love enzymatic amplification. The engineer's approach is to attack this with technology, maybe using a EMCCD camera, confocal microscope, even an electron microscope that can get you down to seeing individual molecules if you look hard enough for them. So this type of approach can be very expensive or very low throughput, but still certainly possible if you throw enough resources at it. So the approach that I'll talk about today is, I think of more as the nanotechnologist approach, where we'd like to increase the signal per molecule using some type of nanoparticle tag. I'll really emphasize today using gold nanoparticles as the tag, but you can also link biochemistry and do single molecule detection using quantum dots as well, photon emitters. If I have time left over at the end, I'll tell you some of the things we're doing with quantum dot tags for single molecule limits of detection for biomolecular assays. And so we're really thinking in the context of approach that can be used to monitor the success or failure of chemotherapy during and shortly after the time where the patient is receiving it, where there aren't really any other good methods. You can't biopsy the person every day. Biomedical imaging methods can see if the tumor is growing or shrinking, but those measurements involve expensive procedures, expensive equipment, not very quantitative. So we want to use a liquid biopsy approach, but with a simple rapid assay, no chemical amplification, and the ability to use a few droplets of test sample, but yet have detection limits that are in the femtomolar range, where there may not be very many molecules to detect from in that very challenging situation. So now I'll get into the engineering part of the lecture, and basically what we're doing is similar to what the genie did in the movie Aladdin, where taking phenomenal cosmic power, concentrating that power into very, very small volume. Where in our case, the phenomenal cosmic power comes from external light sources like LEDs and lasers, and we're seeking to concentrate the energy from those light sources into small volumes represented by nanoparticles like quantum dots or gold nanoparticles like the ones I'm showing here. The tool that we use to do that is an optical metamaterial surface called a photonic crystal. As I'll describe in a minute, a photonic crystal is a surface that we can engineer and build onto glass microscope slides or silicon wafers and do that very inexpensively. But the photonic crystal surface is engineered to capture light from the outside, such as from an LED or laser, and to resonantly concentrate its energy onto a very low surface plane where nanoparticles or molecules that come into contact with that surface confined energy will be exposed to a much greater intensity than they otherwise would. And so in doing so, we can do some very interesting things and develop some new forms of microscopy like I alluded to. Just to show you an idea of the size scale, here's one piece of a photonic crystal wafer. This is a wafer actually originally made on an 8-inch diameter piece of glass, chopped down into die. Here's one around the same size as a US penny. This whole surface is a photonic crystal. If we zoom in with an electron microscope, we can see a photonic crystal surface with a few gold nanorods sprinkled on it. Here's some gold nano urchins, have this spiky surface on the outside. If you look at these nanoparticles, they're very small. They're around the same size as a virus, like around 100 nanometers diameter, not too much bigger than a protein molecule, such as an antibody. And then to further give you scale, here a DNA molecule around 1 nanometer size scale. So the thing that a photonic crystal does that's very interesting from an electromagnetic standpoint is that when you illuminate a photonic crystal with light that matches its resonance coupling condition, the photonic crystal turns into a very high efficiency reflector, but only for a narrow band of wavelengths. And so if we look here at the reflection as a function of wavelength, this specific photonic crystal made on a silicon wafer with SiO2 glass surface, a titanium oxide high refractive index surface, is a very efficient reflector for light in the red part of the spectrum, where at that resonance coupling condition, here looking at a cross-section electromagnetic simulation of the photonic crystal, we see very high electric field intensity around the photonic crystal and extending into the medium like water or air on top of the surface. If we happen to go off at a different resonance, at a different wavelength, slightly off the resonance, now it's like the photonic crystal doesn't exist, and it's just like a regular piece of silicon or a regular piece of glass where electric field intensity is low. So the thing that we do that's like our electromagnetics trick, it's very simple actually, is that we design a photonic crystal to have a resonance at a certain part of the spectrum, here for example around 633 nanometers. What we do is to choose a nanoparticle that has an absorption wavelength that's the same as the resonance wavelength as the photonic crystal. So that if we take that nanoparticle and dunk it into that high electric field region on the photonic crystal, the energy will couple from the photonic crystal into the nano antenna, and that nano antenna will be excited. As I'll show you in just a minute, this effect enables the nano antenna or the nanoparticle to experience much larger, orders of magnitude larger electromagnetic field intensity on its surface than it otherwise would. And so by taking these two things, putting them together, photonic crystal resonance and surface plasmon resonance from a metal nano antenna nanoparticle, we can build very large electromagnetic fields. And so this cooperation is a cooperation between photonic or a dielectric resonator, like a photonic crystal, and a plasmonic metal resonator, like a gold nano antenna. And so we can use electromagnetic simulation tools to conceptually see how well this technique works. And here I'm just showing you a rigorous coupled wave analysis simulation of a gold nanoparticle on a photonic crystal, where the scale bar represents the electric field squared, representing the optical power. So you can see how the electric field surrounding the gold nanoparticle is very high, but confined just around the skin, just around like the very outside surface of that gold nanorod. You can imagine that if we have this gold nanorod, there's electrons inside there, and those electrons are being excited to oscillate back and forth inside that gold nanoparticle. And so that type of electron oscillation will also result in heating, localized heating, and you could even simulate the amount of temperature increase that you would get from a resonantly excited gold nanoparticle on a photonic crystal surface. So that there's some very interesting electromagnetics and physics going on there. One of the things that we observed that was very interesting is that if you have a photonic crystal that ordinarily would be a very efficient reflector, if you put absorbing gold nanoparticles on the photonic crystal, the reflection would go from highly efficient down to less efficient. And it really makes, we call this a new effect, but it really makes sense because the gold nanoparticle is robbing the photonic crystal of some of its resonant energy. And so rather than being an efficient resonant reflector, now the presence of the gold nanoparticle is absorbing energy, stealing it away, so the resonance is quenched partially. And so we can observe this in the aggregate by just looking at the reflection efficiency from the photonic crystal dropping down like this. And so in fact, we studied this recently in a lot of detail to try to quantify the absorption cross-section of gold nanoparticles on photonic crystal surfaces. And so we showed in a nanoletters paper, if we measured carefully the absorption, which is 1 minus the reflection minus the transmission efficiency, so taking very careful measurements of reflection efficiency and transmission efficiency through photonic crystals that have gold nano-urchins on them, we were able to show that effectively the nano-urchin becomes more efficiently absorbing than it otherwise would. And so the basic idea here is that these gold nano-urchins are small. They're around the same size as a protein molecule or a virus, but by enhancing their absorption, by putting them onto a photonic crystal, it makes something small. Looks like it's actually 30 to 50 times bigger than it really is. So we sought to take advantage of that basic electromagnetics effect in the following way. So if we have a photonic crystal surface, it has its resonance at the 625 nanometer wavelength range if we don't put any nanoparticles on it. But now let's say we take a gold nano-urchin and we attach it to the photonic crystal. What we observe is that two things happen. First, the gold nano-urchin has replaced some of the water that used to be there. And so the resonance wavelength of the photonic crystal will shift from a lower value to a larger value. We call that a shift in the peak wavelength value. But also, and most importantly for us, is that the absorption of that gold nanoparticle will rob away some of the photonic crystal's resonance energy, and the reflected intensity will go down. But this reduction in the reflected intensity is very localized only to the place where the gold nanourchin is. If we move away from the gold nanourchin by several micrometers, this effect does not happen. So it's a localized resonance quenching effect. And so that tells us perhaps there's a method we could have microscopy. And so this is where they're leading into the photonic resonator absorption microscope, which is really just a modified bright field microscope with the schematic shown here, where we can take light from a low intensity LED, so no lasers, no blasting with huge amounts of energy needed here. So the LED is focused to a line through a cylindrical lens and hits the photonic crystal from below, illuminates the surface where the illumination light goes in, the reflected light comes back through a microscope objective, and then we feed that light into the entrance slit of an imaging spectrometer. So here's our microscope, spectrometers over here, and then some of this gear over here is meant for doing cell-based assays and experiments where we have a cell incubator built on top of our microscope. So to acquire a PRAM image, we take the light from the LED and it's focused into a line that spans across the photonic crystal. Excuse me. Each point along this line is represented by a resonance reflection spectrum from the photonic crystal. And in one capture from our spectrometer, we gather 512 spectra, each from a different point along this red line. And so by moving our photonic crystal under the line in small increments, in about 30 seconds, we can gather a whole stack of reflection spectra from each position on the photonic crystal surface, and then analyze them for their peak intensity value. And so what's shown here is a PRAM image where each dark spot represents the position of one gold nanourchin, where even though they're small, they have very high contrast. We can see each one very clearly. We can represent them mathematically on a 3D graph as shown here, where it's more intuitive to show them as peaks rather than dips. But keep in mind, each one of these nanoparticles represents a dip, a reduction in the reflected intensity. So we sought to take advantage of this to perform a very high sensitivity and simple assay for microRNA. So now kind of returning to our clinical problem a bit. So let's imagine we can take our photonic crystal and we can prepare its surface with single-strand DNA, just covering the whole surface with this single-strand DNA represented by the sequence of green dots here on the surface. So those are attached there. Now we can take our gold nanourchins and prepare them with a specially engineered nucleic acid molecule that's called a toehold probe. So toehold probes, very interesting technology invented by David Zhang at Harvard, and we've adapted it here for the first time to use nucleic acid toehold probes on nanoparticles for an assay like this. So the toehold probe contains a complementary match to the microRNA sequence we want to detect. So our test sample, like the serum from the cancer patient, has a microRNA with a specific sequence that we want to detect. The toehold probe molecule has a complement to that sequence, but that complement sequence is protected by this nucleic acid that's called the protector. Now, what occurs is when the target microRNA reaches the toehold probe, they have an interaction called strand displacement, where base by base, the target microRNA attaches to the toehold probe and forces the protector to go away. So when the protector goes away, now it's kind of floating around free. This nucleic acid sequence represented here in green that used to be protected by the protector is now exposed. Now we've chosen this sequence of nucleic acids strategically to be a complement to the nucleic acid sequence that we've already prepared on our photonic crystal. And so exposure of the toehold probes on the nanourchins to the test sample, one target microRNA molecule results in activation of one gold nanourchin particle that can now be captured on the photonic crystal surface only when this green sequence is exposed. So basically, this is an assay that converts one target microRNA molecule into one surface-attached gold nanourchin nanoparticle that we can subsequently count with our new form of microscopy. I won't say too much about this because it's really the topic of molecular biology, but these toehold probes are very selective. You can design them using thermodynamic principles to only recognize the target microRNA sequence. In this case, our old friend microRNA 375, and then not any others. And so in this way, each gold nanourchin that's captured reduces the reflected intensity from high to pretty low, as shown here. And so basically the photonic crystal is giving us an enhanced form of contrast for detecting and then counting those gold nanourchin nanoparticles. And so we do some image processing and analysis to turn those images into counts. So we can do some thresholding and so on to count the nanoparticles that we detected. And then we've used these now to characterize our ability to detect microRNA 375 over a range of concentrations. Now, let me draw your attention here. Let's look at the horizontal set of images. Each of these squares is one param image, where the little bright flecks of salt on a black background, each one of those represents one microRNA molecule. So going from lowest concentration of 0.1 femtomolar and then going up by orders of magnitude, we can see how as we go from low numbers to higher numbers of molecules, after here, just showing after a 2-hour incubation, happening at room temperature with no stirring at all, we can cover 5 orders of magnitude dynamic range and have very quantified outcome. Here just shows in bar graph format the same data that I just showed you in the images. What showed to us is that 100 attomolar limits of detection were achievable after a 60-minute incubation time. And so here, if I just focus on the 60-minute mark here, the gray bar represents no target present. This is our non-specific attachment of nanoparticles that land on the surface by gravity. We also had another type of negative control where we had 1 picomolar concentration of microRNA that differed from our target microRNA by only 1 base. And so this represents how selective the toehold probe method is. You can see how low the detection is from a non-target molecule differing from our target molecule by just 1 base, but at a 10,000 times higher concentration. We did some more things in our paper to characterize the selectivity and the placement of a mismatched molecule. So this just really just shows how good the molecular biology is. So I won't spend too much time here on this. And then we finally did some work showing our ability to detect microRNA 375 spiked into serum and still be able to detect down to 100 attomolar concentration and still able to span 5 orders of magnitude dynamic range. We've used the same approach for doing a sandwich assay in the context of detecting a protein biomarker. So same basic idea, except we prepare our nanoparticle with a capturing antibody that we mix in a small plastic test tube with our test sample to gather a protein antigen. And then we incorporated our photonic crystal into a microfluidic device where we could take a single droplet of this test sample, put it in, and then have that test sample run over the photonic crystal while being absorbed with a little bit of slow flow through the use of an absorbing paper pad. So on our photonic crystal, for detecting a protein, we had a reference area that's blocked against nonspecific binding, and an active area that has a secondary antibody that also recognizes our target analyte. And so in this way, we make a sandwich where the top of the sandwich is a gold nanoparticle. The gold nanoparticle, again, represents the capturing and the presence of one analyte molecule. But then we use the same methodology, the same photonic resonator absorption microscopy approach to count and detect the nanoparticles. Here's a schematic of our microfluidic device. There's an absorbing paper pad that draws the samples through at a rate of around 1.3 microliters per minute. So a 40 microliter droplet sample goes through in about 30 minutes. But the user doesn't have to do any other wash steps. Just add the sample in and then wait for the nanoparticles to get accumulated. We used this first in the context of doing a viral load counting assay called an antigen assay for a protein called P24 used to perform HIV viral load measurements. So here I'm just showing you the nanoparticle counts as a function of P24 concentration for the reference region and for the active region. And then we can just subtract one from the other to have the final referenced count of or density of captured gold nanoparticles. We did this first in buffer, and then we more recently did it for P24 assay or P24 protein spiked into human serum, 100% human serum, so kind of a viscous material, but again using the same 40 microliters in 30 minutes, where our limit of detection was around 1 picogram per ml. So I think I've reached 45 minutes, and I want to leave some good time for questions. So I think I'll leave out the final part of my talk. But we've been using the photonic crystal in other ways. So I'll say that we can also use the photonic crystal to enhance light scattering for light that scatters off of gold nanoparticles, viruses, exosomes themselves. And so even without using any labels, we can count things like gold nanoparticles by their enhanced scattering. So this method for enhanced scattering, here we can see gold nanoparticles even down to 10 nanometers size, really small, show up with very nice signal to noise when we can detect them by their scattered signal. We can also use a photonic crystal to excite photon emitters, such as quantum dots. When we design a photonic crystal that has a resonance in the ultraviolet part of the spectrum that excites quantum dots. So we've also done some assays for microRNA using quantum dots as labels. Not the same as the toehold probe method that I mentioned before, but also very selective, where individual quantum dots on the photonic crystal surface can be clearly observed through their enhanced excitation provided by the photonic crystal. And then we can use them in the context of performing assays with single molecule resolution. Here using quantum dots to detect that same microRNA 375 going down to around 0.1 femtomolars. Here we're counting quantum dots rather than gold nanoparticles. So overall, I hope I've convinced you that there's a lot of benefit to be had from finding great collaborators for complementary skills, from the clinical applications to molecular biology, being able to use genomic tools to identify effective biomarkers that can be monitored in a non-invasive way. So we've really embraced this team science approach, and hopefully I shared with you how some of these disciplines come together for some impactful applications. But overall, we're trying to develop, use our engineering skills to develop new technology platforms that can be used to enable frequent, non-invasive, inexpensive assays that can be used to inform life science biology, but also used as diagnostics to inform things like the effectiveness of chemotherapy in the context of prostate cancer, as I showed you here. So thanks so much for your kind attention. I'll conclude here, and I'd be happy to answer questions.
Thank you, Brian. You can see there are some of the questions in the chat section.
Okay, let me find the chat section.
42. Yeah, click on it. You have to scroll up because there are certain questions on the right hand side. You can scroll up. There is a cursor bar.
I'm looking for the cursor.
Yeah, extreme right of this window. No. You have to click on the same chat section.
Sorry.
Yeah. You have to click on the chat section. there's a small strip bar on the right hand side.
Oh, it must be very small. I can't see it. So Manoj, maybe you can help me if you can,
Okay,
if you can
okay.
see them.
What I'll
Then
do is, sure, no problem. What I'll do is, so you can take the questions from the bottom in that case.
okay.
Yeah.
Oh, so have you started efforts to use for the COVID-19 virus? Yes, yeah, we do have a lot of irons in the fire now for assays for detecting intact viruses. Using that scattering approach, we can actually capture and then image individual coronaviruses. And so we're preparing some work on that. We're also using some methods for that, just as I mentioned, for detecting coronavirus antibody and coronavirus antigen that are very similar to that P24 assay that I just showed you. So we're interested in like a point of care, single step, very quantitative assay that would overcome some of the limitations of the lateral flow test strips that don't have very high accuracy or quantitation. Yeah, but I wanted to get away from coronavirus for a day. I don't know. We've been just deluged in coronavirus here in our research.
True. Pick up the next one by M. Chandra.
How crystal sensors can be used in cardiac study and diagnosis. So also interesting question. And in our research, we haven't gotten into cardiac biomarkers yet, even though I'm aware that there are protein biomarkers for cardiac health, and then also genomic ones as well. And so the same methodology that I showed you, once you've identified your molecule, then next thing is to identify, if you're using a sandwich assay, antibodies for selectively capturing it, or if it's a nucleic acid biomarker, designing a probe for a circulating DNA or circulating RNA that's a known biomarker for cardiac disease. I've been finding that there's just almost any disease you can name has protein biomarkers and also nucleic acid biomarkers associated with it. And we've been extending our work into things like pre-eclampsia diagnostics and maternal health and nutrition. There are even genomic biomarkers associated with anxiety that chronic stress can also cause changes in gene expression that show up in biomarkers you can find in the circulation. So it's very interesting. So here's a question: why only gold has been used? Are there other materials that can enhance the sensitivity? We chose gold specifically for a couple of reasons. One is that it's pretty inert chemically. And also there's a lot of very nice surface chemistry methods that can be used to covalently attach nucleic acid molecules and antibody molecules to gold. Also, it's possible to use other metals like copper or silver, but silver will oxidize in aqueous solutions, and so they don't have great longevity. We've also been doing some really interesting work with making magnetic plasmonic nanoparticles that are comprised of gold, but also iron and nickel, that they can be attracted and manipulated using externally applied magnetic fields. So the main criteria for us is that the nanoparticles need to be absorbing at the wavelength of the photonic crystal's resonance. Then after that, we can use many materials or combinations of materials. So a question from Manesh Chanda. Can SiO2 immobilize the DNA RNA perfectly? Is there any other oxide layers for better immobilization? So I didn't describe in enough detail, I think, but the photonic crystal requires a high refractive index material to give it its optical confinement waveguiding property. And for that, we use titanium oxide predominantly because it has a high refractive index. However, you could also use other high refractive index materials in place of titanium oxide. So silicon nitride is effective, hafnium oxide can be used. Sometimes surface chemistry methods are more friendly for silicon oxide than titanium oxide. So sometimes we'll coat our titanium oxide with a few nanometers of silicon oxide so that we can use surface chemistry methods that are already compatible with glass surfaces. So there are a lot of things you can do to play around with the final surface, the upper exposed surface, but you do need to have a high refractive index material to create the photonic crystal. Okay, so here's another question. Does the orbitals of the gold atom have temperature difference? Oh, that I don't know. Yeah, and so here, I mean, we're really just using, the gold's ability has many free electrons, and those free electrons escape from their atom of origin, and those electrons are free to resonate around inside the gold nanoparticle. So I think the free electrons aren't so associated with the atom. I don't usually think of them for their orbital. I hope that answer makes sense. Another question, how are the antigens immobilized on the gold nanoparticle or on the photonic crystal surface? Yeah, so excellent question. So capturing molecules need to be attached to the gold nanoparticle and to the photonic crystal by covalent chemical bonds so that they don't just simply wash away. There are many approaches that can be used to make that kind of covalent linkage. In our case, we're using something called a silane layer, where the silane is a silicon-containing molecule that makes a covalent bond to the photonic crystal. But then the silane molecule has another functional group. It's like a bifunctional linker that can make a covalent bond to an antibody or to a nucleic acid molecule. So I might, rather than, it's hard to describe details by waving my hands. I might refer you to the paper to look at the exact details of how we did it. But we describe our surface chemistry approach in a lot of detail. So another question, can the photonic crystal sensor be useful for label-free detection of procalcitonin, small protein and biomarker for infection, concentrations of 1 picogram per ml? Yeah, so I'm not very familiar with procalcitonin, and so my answer is I think it's possible, but I'm not sure. So for label-free detection, you'd put a capturing molecule on the photonic crystal and then measure changes in the reflected wavelength of the photonic crystal as the capturing molecule captures the target. And so one key is having a high affinity antibody molecule. And then I think picogram per ml may be around the limits of detection for detecting a molecule around that size with high signal to noise. But I'm not aware of someone having done that exact study.
Okay, Brian, there are certain, we are 3 to 4 questions more. If you're not able to scroll down, can you just exit from this chat section and click again? Yeah, can you click on the chat section again?
Oh, here, I'll stop presenting.
Yeah, and now you go, so now you click on the chat section.
Okay. I still can't scroll. I wonder what's going on there. I don't have the little bar.
There's a very small, thin scroll bar on the right-hand side. You can scroll up and scroll down.
No, I'm looking for it, but I can't find it.
Oh, you're not able to find. No problem. You take the questions from the last in that case. Okay. So which question you are able to see?
Sorry, I'm looking
no
right
problem.
at the spot that you're guiding me to, but I just don't have a scroll bar there.
No problem. No problem. Which question you are able to see right now?
Now I just kind of restarted. Now I think I'm at the bottom. It says.
There is Bhumika
Oh no, these
Sharma.
are questions from before.
Yeah. You just have to.
Okay, here's another question. How to give a virus proper environment under test so that it can be traced to some nano quantum dots using some metamaterial? I'm not sure I understand that question. Let me move to.
Yeah, you can take up the next one. Yeah.
So working on perovskite nanomaterials, are they suitable for this purpose? So Perhaps I wish you were here. I could ask you more clearly what you mean. So in our case, we're using a photonic metamaterial, like the periodic structure, to generate these optical resonances. And I don't know if you can do that with perovskite materials, or if you can make like a surface out of them, or whether your nanomaterials are more like particles. Gold nanoparticles aligned in a one-dimensional metamaterial provided an almost linear sensing response. However, a one-dimensional grating produces a broadband zero diffraction order beam and senses a single strain direction. Can 2D gratings using photonic crystals sense in multiple directions? Yeah, that's a really excellent question. So you notice here that in all the work that I shared with you today, they were one-dimensional photonic crystals. They're only periodic in one direction. But you can also make a two-dimensional photonic crystal where you have a periodic array of holes or a periodic array of posts. The advantage of the two-dimensional photonic crystal is that the resonance is not selective on the polarization of the light that's applied. And it's not selective to the polarization, like the orientation of nanoparticles like nanorods. So notice that like the nanorods have two potential orientations. What we found is that nanorods that are oriented with their long axis along the electric field direction are really good at exchanging energy with the photonic crystal. But if you rotate them 90 degrees, they're much poorer at exchanging energy. So that's one reason why we developed a preference for nanoparticles with spherical symmetry, like the nano-urchins, because their orientation no longer matters. But there actually are very interesting things between the orientation of the electric field polarization on a photonic crystal and the polarization direction of molecules and nanoparticles that you can kind of see interesting physics and also potentially exploit them too. So that's a really good question. Have any side effects on the body for using quantum dots and gold nanoparticles to diagnose cancer? So in this case, we're not putting any gold nanoparticles or quantum dots into the person. So these are all assays where we take the test sample out of the person and then do the test in a microfluidic cartridge, outside. But you're correct that quantum dots do have concerns associated with them in terms of their toxicity, especially ones made out of like materials like cadmium and selenium. And so there are people seeking to make quantum dots out of less toxic materials for that reason. And then gold nanoparticles, I'm not aware of their toxicity. The gold tends to be more inert as an atomic material, but I guess I would be concerned where they end up after you put them in the body, whether they aggregate in the liver or whether they actually end up getting expelled through the urine or something. But I'm even aware of gold nanoparticles being used as a therapy in diseases like arthritis. And so I'm sure there have been some safety studies done with gold nanoparticles in the body.
Brian, you can take last two questions if there are any, and then we will close.
Oh, okay. I'm looking. I still can't scroll.
Oh, you still cannot scroll. Okay, okay.
Right.
That's actually on the chat section where the last. Okay, I think if you're not able to trace the things, what I'll do is I'll post some of these questions which I can see over here to you via mail, and maybe if you can just respond, then I'll pass it on to the attendees in that case subsequently.
Okay. Well, I see one more question.
You're able to see one more? Good.
So which materials are suitable for quantum dots, and what are the criteria for selection? So our quantum dots are ones that we've purchased from commercial source. All the quantum dots that we choose have their emission in the visible part of the spectrum. So we prefer those because we can detect them with our imaging system. But then they all have the same absorption spectra where they're excited in the ultraviolet. We're choosing quantum dots that emit at distinct wavelength bands. What I didn't show you is work we're doing with multiplexed assays using different quantum dot emission colors for different biomarker molecules, where we choose the quantum dot emission wavelengths strategically so that they don't overlap with each other, so we can differentiate them one at a time.
Thanks, Brian. Thanks a lot. And on behalf of National Academy of Sciences, India, Delhi chapter, and the college itself, I would like to thank you, and I would like to thank all the attendees who have spent nearly more than 1 hour 30 minutes, because they joined somewhere around 7 p.m. India time.
Oh.
And this is dinner time, I know, in India. And you perhaps, because it's 9 a.m., I suppose you joined at 8:45 a.m. That must be a breakfast time for you. And you have so that's great.
To meet you, Manoj, and really grateful for everyone taking the time to listen to my talk today. And I wish everyone a wonderful evening, and just take care of yourselves.
Thank you very much.